Thanks a lot. Always great to be here at this meeting and to see all the progress that's being made in the different therapies, be it ex vivo, where we're seeing fantastic results from CAR-T, CAR-M, and also in vivo therapies like we just heard it. I'd like to take you to, well, maybe one step back, maybe one step forward, actually to the technologies, how we can most efficiently make the viral vectors that are needed for these gene therapies. Before I dive into that, I'm very happy to share with you the not so new news that we are no longer a standalone company, but that we are now part of Cytiva since last year, October. And this also opens up a whole new set of possibilities for our business and for what we're doing. So as many of you know, probably know, Paul Life Sciences and Cytiva are currently coming together and offering between them basically the equipment and products for viral vector production throughout the upstream and downstream process. What Cevec has been adding to this is actually the biology. So bringing in cell lines that will help production of viral vectors. And I would like to focus this talk on two technologies. One is our EleVector technology that we have invented and established and also been partnering out over the last years, which is for AAV vector production, and I'll lead you through the process and how we can actually help to make this process more robust, much more scalable, more cost efficient. And the second one, to which I will dedicate a few minutes at the end of the talk, is our CAP-AD platform for the safe production of adenoviral vectors. So we've just heard how well transient transfection for AAV production can work. When we set out to develop a system, our credo was we need to make it as easy as antibody production. It has to be done by stable cell lines, and we still think this is the way to go. When we started to establish the technology, people told us, you're crazy, that's never going to work. I'm not good at taking advice, so which was good in this instance, so we made the technology work. So our EleVector platform actually integrates all the components that are required for AAV production into a stable producer cell line. That means the rep genes, the helper genes, the CAP, and the ITR flank therapeutic gene are all stably integrated into the cells. This is an inducible system, since some of these genes are toxic, so you couldn't just make a straight stable cell line. This has also been the tricky part, but we have it nicely under control, so we have a tightly controlled system where we can generate the stable cells. They will then not require any further transfection or any helpervirus infection. They just go straight into GMP banking like you know it from monoclonal antibodies or from proteins. And then when you go to production, you expand the cells production volumes. That can be the 200 liters we've done. We didn't go further for cost reasons, but that went very nicely and smoothly, so there's no limit to go up to higher bioreactors, just standard stir tank bioreactors. And we initially generated this system on our proprietary amniocyte cell line, but we realized everybody's using HEK293 cells for making AAV, so we should also have an HEK293 version of the system. This is what we have been establishing over the last few years. So we took the standard HECC, HEK cells, adherently growing. We suspension adapted them to serum-free media in chemically defined media, single-cell cloned them, so there's now a nicely characterized single-cell clone growing in single-cell suspension. The cells have been fully documented and banked, and they serve as the basis for the EleVector HEK platform. What I should also mention very briefly here, we're hoping to launch next year an improved version of these cells that we have modified, and we do have the cells already, but we need to sort of productize them yet, that we have modified to actually greatly reduce the encapsulation of host cell DNA. That's an impurity that you cannot purify away, and is of concern if you're actually applying larger doses to patients. So these cells will basically get rid of that problem. They should become available next year. We started with the cell line to also establish the vector platform on them, and we have successfully done that. It looks as if we're getting at least as good results from the HEK than from the CAP cells. And obviously, what we're doing for each individual product, for each individual partner and client, is we generate the producer cells, because the capsid is going to be specific, depending on your targeting profile, on your safety profile. Very often our clients just come with their proprietary capsid, and of course the therapeutic gene is proprietary to the client. So we start with an alpha cell line that has everything except capsid and GOI. We do the cell line development, basically a stable transfection of the two constructs to generate a polyclonal pool population that from those we already generate preliminary material that we can analyze. That also gives you an indication on how well your construct performs on this platform, and then of course go into single cell cloning, standard single cell deposition by cell printing, single cell analysis, proof of single cell or proof of clonality, and then analyzing the clones for productivity and also in the initial screen full empty ratio. We then go further to an AMBER system to do initial process development for the most promising clones and pick the top clone. For the HEK platform, that's just one example of a very first proof of concept cell line where we did just a very limited process development, ended up with 4 × 10 to the 10th Vg per mil, so a decent titer, nothing to write home about, but we are okay with it. We've now taken these cells into other projects where we're actually seeing more than 10 to the 11th already at the polyclonal level. The single cell clones usually go up by factor 5 to 10 from the polyclonal pool. For the CAP cells, we have also done further process development, and this is just an example where the pool started at 10 to the 10th Vg per mil. We then did the single cell cloning, gaining an order of magnitude in productivity, and then did process intensification, something that you can very nicely do with a stable cell. That's tricky to do with transient transfection. And here, gained another order of magnitude. So we ended up with 3 × 10 to the 12th Vg per mil, 30 to 40% full particles in this particular case. And you have a very reproducible process here. Since you're dealing with a stable cell line, you don't have any inconsistencies in transfection efficiency material that you're adding. So with this platform, what we're doing, as you can imagine, is we are not developing our own products. We are partnering with developers to generate their producer cells for them, to characterize them, and then hand them over for production under a license from us. As promised, I wanted to share 2 slides with you on our CAP-AD platform, which we have developed for production of safe adenoviral vectors. Adenoviral vectors have got a lot of traction actually during or after the pandemic or during the pandemic with the development of the adenoviral-based vaccines. So I think this has sparked a lot of new projects for vaccines and other applications for adenoviral vectors. And some inherent risk in adenoviral vector production is actually the formation of RCA, so replication-competent adenovirus. It happens from time to time when you produce on an HEK cell, just due to the fact that the HEK cell just contains a fragment of the Ad5 genome. And this fragment has overlapping sequences with the adenoviral vector. So that means if you have an E1 deleted adenoviral vector, for example, you can, in rare instances, have homologous recombination between the vector and the Ad genome in the HEK cells, leading to RCA. It's a very rare event, but you do find it in about 5 to 500 cases if you analyze 5 to the 10th particles, 5 times 10 to the 10th particles. The CAP cells were actually by design modified to avoid this. So they don't have two homology arms. There's only one very small homology region. So this avoids any homologous recombination. So when we analyze these 5 times 10 to the 10th VP, this is the standard analysis, basically the threshold you have to meet. We don't find replication-competent adenoviral particles. And of course, productivity has to be maintained. So when we compare it, for example, to 293F cells, just transducing or infecting the CAP cells or the 293F cells with an adenoviral vector, we end up with the same yields and the same amount of transducing units from these cells. This system is also based on suspension cells, serum-free, chemically defined, fully documented cell lines with a DMF filed with the FDA. And in this case, there is no cell line development involved because that cell is already the producer cell. So all you need to do is take GMP vials from these cells and infect them with your adenovirus, or do a transfection of a rescue of the adenoviral plasmid to generate a new safe adenovirus. Again, here there's no service component. It's a pure licensing model. So our partners and clients come to us, and we will out-license the cells to them. And now, since we're part of Cytiva, what we'll also be able to offer is actually the process development. So we can give you the cells, including a fully developed process for your specific vector. And with this, I think, we even have 3 minutes for questions.